the talk I'm going to have today is kind of hopefully something that has some practicality there that will tie to what you're doing. So we've had Walter here showing you that we have this phenotypic data that's been collected from our potato diversity panel, and we have the phenotypic data. What's also sitting there is our SNP data from the 8,300 SNPs, and Robin's postdoc Candy Hansey has been started the analysis on that data. So she has the 8,300 SNPs for the diversity panel. She's going to be working with the phenotypic data, and we're going to try to start doing some association analysis on that. So it's kind of work in progress there, and so this really is a team effort to move forward with this. So what I want to talk about today is SNP-based linkage maps, and kind of how do we get to the linkage analysis. And then I'll go a little past that. I think the point here to make is, we have our SOLCAP team of scientists, and this effort to create the SNP-based map is a team effort between Robin, myself, my technician Joe Coombs, who's sitting here, Kim Felcher, a postdoc in my lab, Dan Zarka, a postdoc, and Candy Hansey, all kind of working together. And so what we're giving you here today is we've gone through all this process to create this SNP map. And so this is, if you're going to generate a map, how do you go through, take this data, and filter it down into something useful? So we've got kind of a procedure for you to follow, so you don't have to reinvent this process. Okay. So when SOLCAP started, we had one mapping population that we considered as part of our purview, that we were going to do the SNP genotyping, get the phenotypic data to see if we can identify some QTLs, and that population is the Premier and Rio Grande, and Walter presented data on that. Our other purview was that to open it up to the community to submit populations that we would then consider that we thought were of value to the community, that we would SNP genotype, and then you would have your phenotypic data, and you can move forward with that. And so on this list here are a number of other populations that have been genotyped by the SOLCAP group. Plus, I actually included some of the ones that I've done myself. So we have Atlantic by Superior, which is supposedly a chip processing cross. We've got B1829-5 by Atlantic, which is out of Craig Yancho's population out of North Carolina. We have a population here, a chipping population, Wisconsin 2310, which has been named Tundra by Kalkaska. I'm going to jump over this one for a moment. We have another cross here, tetraploid population that's a cross between Jacqueline Lee and MSG 227 for late blight resistance. And then Walter has a tetraploid population, Juanita by Pike, which offers us also some chip processing. At the diploid level, we have Shelley Jansky's population that's being genotyped, and we have two other diploid populations. And then lastly, we have a set of 48 clones from the International Potato Center that have been SNP genotyped. And I think that's going to provide some interesting germplasm comparisons between the U.S. germplasm and the South American germplasm that they're working with. What I'm going to focus on today is these two diploid mapping populations. And so you can see Robin's talked about DM, DM 1-3, and it's kind of amazing to kind of step back and look and say, I remember when Richard was developing his monoploids for the purpose of trying to come up with a breeding system that would reconstruct heterozygous tetraploids. And out of that work comes this doubled monoploid that actually has ended up helping us for a whole different purpose, help for the sequencing, and I think it also helps for our mapping purposes. So to move forward with the mapping, we thought one of the simplest things to do is to start crossing DM to some other diploid lines so that we only have one parent segregating to simplify the genetic analysis. So we initially developed a mapping population for DM by 84 SD22, which is a Chacoense tuberosum hybrid. And then after the data came off the SNP genotyping, we were looking at it and we said, maybe we really need to have a second population. And so we contacted Richard. He had the cross already in hand, DM by RH. He sent us the DNA, and we were able to SNP genotype that. And so having two populations to work with, I think, is more informative than having just the single population to move forward. And so what we want to do today is kind of go through the process of SNP genotyping a population. And so we need to create a mapping population. So here's the two that we're working with. We kept the population size to a plate size. We used 92 progeny plus the parents, and this is just showing you, Robin showed you the tubers of DM earlier. They're kind of an elongated tuber with some coloration, and then 84 SD22 is kind of mostly a white skin tubers with white flesh. And interestingly, the progeny kind of ended up being purple and red with even flesh color, purple and red flesh color in them. So my talk here is we're using the Illumina Infinium platform. That's what SolCap has worked with. So this is not an advertisement for the Illumina system. There's many platforms that we can use, but we're working with the Illumina platform, and it's worked out very well for us. Okay, so if you create your mapping population, what you need to do is purchase Illumina Infinium potato SNP chips. The window for ordering those is actually passed, and there's been questions, that have come to me by email. Hey, is Illumina going to be making any more potato SNP chips? And so I think the answer is yes, they will make more SNP chips if there's enough of an interest. So if you think that you want to invest in some SNP genotyping, why don't you let me know so then I can kind of collect those numbers to see if we're getting to a point where Illumina is interested. We need to have kind of a minimum order so that it keeps the price per genotype sample at a reasonable cost. The original round was $85 a sample, what it worked out to, and I think that has been a reasonable place to be. It seems like it'd be nice to get a little cheaper, but we'll go with that. Okay. Let me back up there. And so what we're going to end up doing is we need to isolate high quality DNA, and we need to quantify that. And this has been a little bit of a process. The procedure for the SNP genotyping is a 3-day process, and to me, that just is amazing that we can analyze 8,300 genetic markers in 3 days in a population of 192 if being. So the system's set up to run 192 samples in 3 days. And Dan Zarka is the postdoc in my lab that's been running all the SNP genotyping. He's been doing it on potato, done some on tomato, and he's also doing it for the rose breed program, which is doing cherries and peaches right now. And I have to put a plug in for the Illumina chip system, is that the data coming off it has been excellent. We haven't had any failures, and so the whole process of generating markers, to me, is taken out of that the work. And so if you invest in these chips, what you can do is invest more time in the genetic analysis and the phenotypic analysis. So I always feel like the old time of the grad students spending all the time in the lab generating markers is kind of past in a way. Okay, so it's this 3-day procedure, and that's the truth. So then we get that the data files. They're imported into a program called Genome Studio. So if you're going to work with the SNP data, you need this software from them, and then you can initiate your mapping. And so here is kind of your key comments that we've summarized. And after all the work that we put in, this is the way that that some of the things to think about so that you do things correctly. One is you've got to input your reps with unique names so that you have so that Genome Studio reads your data correctly. So that we have a kind of a standardized system of looking at the SNP calls. We want to always export it as a design strand. I'll come back to that. And then what we want to do is remove all questionable SNPs, bad SNPs, SNPs that map to multiple places, missing data. So that, these are things that you need to work through. You need to check the parent replications to make sure that the genotype calls agree. And so you can check, and so for example, in our case, DM is supposed to be homozygous. And so if the SNP isn't homozygous, maybe we want to pull that out. Check the parents for ability to segregate in the population. We can also need to look at the if there's skewed segregation and what's going on. And so the Genome Studio is going to be important to that. You're going to make your map, and then go back and look at your skewed markers. So that's kind of like the process that we feel that you need to go through. Okay, here's a little primer on the SNP chip as it finally stands. So it was originally supposed to be a 10K chip, and then when you put when we submitted our SNPs, they only actually, I think, used 9100 because some of the SNPs required 2 beads rather than a single bead, and so and then of those 9100, they have a certain percentage that didn't work. So we actually had 8300 that actually worked on the chip. Now, of those 8300 SNPs, we didn't just randomly put them on. What we tried to do was focus on SNPs that were in candidate genes. If you remember, there were calls to put out, people sent in their favorite candidate genes. Robin collected all that information, and then we focused on SNPs within those candidate genes, and many of those are included on the SNP chip. In addition, I think this was really nice, is that Robin went in and found found SNPs that were in already published genetic markers. So that gives us a little bit of reference there. Then this is, I think, the power of having the sequence is that the rest of the SNPs, based upon where they are in the genome, were chosen to maximize the coverage. So when it was all said and done, about 650 megabases of the genome was covered with by our SNPs. So, and if you remember, Robin talked about that there were 727 megabases in the DM genome. That was a sequence. I think we're in a pretty good place. Okay. So is 8300 SNPs good enough? I think so. There's other colleagues that are working with soybean and corn and all that. My colleague at MSU that works on soybeans, they have a 55K chip, and but I don't think we need that many markers at this time. It would be cost prohibitive. So I think SolCap is invested in already SNP genotyping about 1,600 genotypes with the SNP chip, and then other programs have been purchasing chips and getting that done. So at this point, over 6,000 genotypes have been ordered, or chips for about 6,000 genotypes have been ordered worldwide. So I don't know where, who has those chips or whatever, but we have a large number in the U.S., but over in Europe, they've invested in that, so I think that this has been a success. Okay, so kind of getting back to the procedure here is that I think what we need to have is the high quality DNA, and it's kind of funny that something that's been around for decades is kind of one of the bugaboos in the whole process is, and this doesn't just go for potatoes, this goes for tomatoes too, is the simple process of isolating high-quality DNA at a concentration good enough has been some of our holdups in the process. And so we've been recommending either a QIAGEN or CTAB method, but importantly is that Illumina says that they quantify their DNA using a picogreen assay, and that you need 50. Okay, hold on. And we need to adjust the concentration of 50 nanograms per microliter, and so we need about 15 microliters to make sure we have enough to do the analysis. And Dan Zarka says if it comes in 96 well plates with self-adhesive foil, seal and cover would be the best way to receive that. Okay, so we've received it in many different ways, but the key is this concentration. And if you're using these chips, each chip holds 24 genotypes, and so each chip costs over $2,000. So you want to make sure that you do it right. And so we've tried not to skimp on the DNA quality and concentration. I will admit that we've put samples on with less than 50 nanograms per microliter. Dan has taken it a little bit lower than that, and it has worked out. But it's nice to use this as a standard, okay? And so I'll just step back there. I put in there, thank you, Dan Zarka, because every chip that we've done has worked right now. I can't imagine what it would be like if we had some failures. But we've been getting excellent data, and Dan Zarka has been taking on the responsibility of doing the genotyping, and I'm afraid to move it to anybody else because we've been so successful at that. And so it's truly a 3-day procedure. Everybody always likes to make something seem a little better than it really is, and so I would always tell people, oh, it's about a week. But after it's really gone through it, it's actually been about a 3-day procedure, and it's a microarray based system that we use. And so what comes out of this is an intensity data file. Hold on, let me see if and what I'm not seeing here is I can't remember, but we have each of the wells on that chip, the 24 wells, there's about 250,000 beads, and of those, each of those beads are replicated 15 to 30 times. So whenever there's a call made on this eye scan, when it's the laser that's reading it, what it's doing is taking an average of those 15 to 30 bead reads, and so you're getting a very good call if your beads are read correctly. So it's unlike a gel where you load a sample on, you got a well, and you got a band, or you don't have a band, or you think you have a band. I think this is actually more reliable data that comes off the scanner that's read. Okay, so now we have the data files in hand, and so what Dan will do is he'll import them into Genome Studio. So you need the intensity data file, and what it is, it's a light intensity or color that's being read. There's a sample sheet that he creates so that we can correlate the data with the intensity data with what was loaded on, and then there's a speed manifest file that comes from Illumina that gives the location of all the beads and what SNPs are there. So it's that data file that needs to be aligned with the intensity file to give the proper reads. So now he's taken these 3 data files, imported them into Genome Studio. And so this is a screenshot of Genome Studio here. And so over here, where I have this circle, is what you're not seeing is if we were to expand this window of information, all the way going out to the right would be all the progeny. So, and if this is our diploid cross, there should be 92 progeny plus the parental lines on there that are continued down to the right that we could scroll over and look at. And then over here, what you have in this column are all the 8,300 different SNPs. And so you can kind of highlight a single SNP and look at the data across all the progeny there. And here you can see is the call that the auto call that was made by Genome Studio. Okay, and so here we got the homozygote, BB, homozygote AA, and so on. Over here is actually the graph for you to view what actually came off the eye scan. And so you have this theta value, which you see over here, theta, and you got the theta values here, and you have this this normalized R value that's over here, and so that's plotted out on the graph here. And so over in this section here, is you see them highlighted in blue. And so what you can do now is you can say, oh, I want to look at the parents, or I want to look at one of the progeny or multiple progeny. And so you can highlight them here, choose the color, and then look up here and see what you see. So the ones in yellow here are the ones that are highlighted. So these are the parents. The DM, we have 4 reps of that, and the RH, we have 2 reps. And so you can see them highlighted in yellow. So you can choose the colors and all this. So it's a pretty nice program. And so you want to be able to manipulate, move around in this program, because you're going to come back and want to look at this information after you've collected some of your segregation data. So I think, so I'm going to move on and kind of just remember that you can do a lot of things in this program, but what we're going to do now is we're going to export this data into something so that we can move it into a mapping program. Okay, and so they have a tool called Report Wizard, and so you go into that, and what you do is just select matrix. Remember earlier I talked about design strand, so you select the design strand as on which to use as your reference call, and then remove we don't need to include the gen call score in this report. So it's calling it a report, and what you're going to do is it's creating a text file that we can then import into Excel. Okay. So first thing we want to do is remove bad and questionable SNPs. So here's some good SNPs, and so in other words, if these were our homozygous parents and these are our heterozygotes, you can see that what we're looking for are ones that are not near the x-axis, and that they're distributed or tightly clustered within the different regions. So over here would be our one homozygous class BB, and over here is our homozygous class AA, and then our heterozygote class in the middle. Over here you can see a one-to-one segregation between the progeny there when you have one heterozygous parent. So here's some examples of bad SNPs for you. So you can see here we just don't have any tight clustering. It's just a scattering of dots all over the place. Here, we're not seeing any type of segregation going on for the progeny or parents. Here again, just a cloud, right? And then here, I was talking about that when they're close to the bottom here, or close to the x-axis, that is kind of questionable whether we got a good call on that. So there's many more bad calls that we could consider. Okay, so Candy Hansey, Robin's technician, has kind of gone through the diversity panel and the Rust mapping population and the Diploid mapping population, and then went once each SNP by SNP over through the 8,300 SNPs to make her calls. Oops, sorry, on that. And just for you to see, so of those 8,300, she felt that 7,412 were good calls. So this is over a broad, germplasm base in which she's basing this information. And then you can see the other parts here, the questionable segregation, questionable bad segregation, and then here's a call rate for the good markers, how over 90% of them have, or actually over 7,000 of them have a 90% good call rate. Okay, so these are the ones that we want to work with. So we filter out those 900 bad SNPs. Okay, it's amazing. We're throwing away 900 genetic markers. Okay, so these are the steps that we want to move through here. Is we're going to walk through this process here. So you can go to this website here, grab the data file, and you can remove the bad questionable SNPs. So you can go in there. There's a list of them there. You can align it with your data that came off. You can also remove the SNPs that map to multiple locations in the superscaffold. So you want to filter those out again in the same file. We're going to remove the parental genotypes that do not agree across replications. That's kind of logical. We want to remove SNPs if we don't have a genotype for one of the parents. We've got so many to work with. Why would you keep one in if there was a no call on the parents? We also then, this was kind of a subjective, is if we felt there was too much missing data for a specific SNP, we decided to exclude it from the analysis. And of course, if the parents are both homozygous, we excluded it and removed unexpected genotypes. And so if DM showed up as heterozygous, that's kind of a pretty good clue that wasn't good. We also removed ones that had highly distorted segregation or no segregation. Now, some of the ones I've got listed here are ones that we're going to walk through in join map. Oops, sorry. Is we're going to remove this co-segregating SNPs. And also remove any individuals that are identical at all loci. We're going to calculate the groupings. We're going to select groups for mapping, determine the marker order. Then we can actually go back and add in the co-segregating SNPs, and we can compare these to the pseudo molecule that Robin talked about, and we can determine the chromosome that these linkage groups correspond with, and also calculate the map size. So the question is, what's the rationale to remove the co-segregating SNPs? For the simplicity of running join map, they're going to choose one for each recombination bin, and so we don't need to have all them, the SNPs that map to the same spot. I mean, they're because of recombination, so it would just kind of make the map overpopulated with markers that are not really informative at this point.
I would argue that they
Yeah. So we put them back in after we do, but we don't want to lose them. But to run the map, we
and then you might be discovering that there are different phases of the train. So you need to
well, yeah, you can use any of the ones that you want that co-segregate. Walter?
Why are you taking up ones that have segregation distortion? My experience is Diploid now. Lots of markers are seriously distorted.
Right. So Walter's asking us why are we taking out the distorted one. I probably shouldn't. It's probably we're taking out actually the ones that are distorted, and the ones we actually removed were the ones that, even though the one parent was heterozygous and the other was homozygous, all the progeny were homozygous. You know, they or all heterozygous. So there was like a complete distortion. So it's hard to read those. But yes, we kept in very highly skewed lines. And I'll point that out to you here. Okay, so here's the filtering process. So we have the two populations, the DM by 84 SD22 and the DM by RH. And so you can see the process here with the first filtering, removing questionable and bad SNPs, removing SNPs with more than 10 missing calls. That was kind of our arbitrary call since we're working with 92 progeny. Remove SNPs with inconsistent parent calls, map to multiple pseudomolecules. So that took us from 7,666 down to 6971 and 7149 for the two populations. Then we removed DM and 84SD22 no calls. And so here's the big jump is when we remove the number of SNPs that were homozygous, same or different within so there's really no segregation is what that's saying. So there's no informative segregation. So those really are the key there. And then we removed the heterozygous DM SNPs, which, you know, in this case weren't many. And then remove SNPs with no segregation. Those are the distorted ones, Walter. And then remove the. So now you can see here's the numbers. So in DM by 84 SD22 we had almost 2,500 segregating SNPs in that diploid cross. And in DM by RH, we had almost 2,000. So that's a good number of markers to work with in those populations. But when we did the linkage, analysis, of those, only 637 in the DM by 84, and only 958 in the DM by RH were unique. Or segregated uniquely, okay? And so those were then selected for generating the map, but then we can add those back in for the final map, okay? So we're using Join Map 4.0 for our linkage analysis. Somebody could use a different mapping program, but this is the one that we used. We had a site license for it at Michigan State. So we've now taken going back to the Genome Studio, we created that report file. This text file was imported into Excel, and this is what you see here without our little text up here, data exported from Genome Studio. That's our little note to you there. So this is a screenshot of the Excel sheet with the information. So you can see we've got 96 samples, 92 progeny and plus the parents, and then 8,303 SNPs. So then there's a lot of data filtering that goes on here. Those things that I was working through. to remove the non-segregating SNPs, the ones that the calls that don't make sense. So what we've done is taken the file here, and now we've got the two parents lined up in the first couple columns. We have the SNP marker and then the progeny going across. You can see all the calls. Up here we gave the name for all the different progeny so that we can distinguish them. And now what we need to do is take this and format it for join map. And so what we have to do is actually change the SNP name. There's a limitation in join map on how many characters the SNP your locus name can be. So we reduce that down. You have to put the type of segregation you're expecting. In this case, it's LM by LL. That's just something that you work with. And then here are and you have to convert from the SNP calls of A's and B's to these LL versus LM. So just some data manipulation that goes on. So I'll say is that what we want to do is sometime in the fall is create some type of workshop, hands-on workshop, where people that are interested, there's a lot of people that have generated populations and collected the SNP data, and what we can do is have a workshop where we can all meet at one place and kind of help you work through some of these things if you feel you need the help. Okay, so we've got now the data set ready to go into join map, and so you can see here the individuals are up here. It only says X, but because the file's been collapsed down, or the columns have been collapsed down, you don't actually see the name there. We got the segregation type, as I mentioned earlier. We've got the locus name. Now you can, call them whatever you want. And we actually at one time was noting in here which ones we saw had skewed segregation, so that we could see how they mapped out, or we had them kind of denoted whether they were candidate genes or genetic markers, just to kind of see where they were all kind of falling out. So you can play around with this, and right now these are the SNP names we're using, but you could put some other parameters into that if you wanted to. So then here's the array size. So we ended up here. This is the DM by 84 SD22. So there's 2,474 markers, 92 individuals, and then the population type that we use is cross-pollinated. So these are just some of the things that we've worked through to get that set up. So now you've run that in join map, and one of the first things that we get is we get some segregation data, and so here's our SNPs and the progeny are going across, and over here on the end here, you can see over here you can see the chi-square value. You can see the segregation data over here. So in the ones that we have highlighted in blue, all these were the ones that had a heterozygous parent and a homozygous parent. We expected a 1-to-1 segregation, and we had 92 to 0. So we removed those. However, we kept in you can see the skewed ones here, 79 to 7, 81 to 11. We didn't want to remove them at this point yet. Because we know if you go into literature that there's a lot of skewed segregation in the diploid population, especially when there's wild species involved. So another way that you can look at the data is the individual genotypic frequency. So you can see now the progeny here, and going across, what it's doing is seeing how many fell into the different homozygous or heterozygous SNP category. And you can see in this last column here, I think, is the number of missed call SNPs or whatever, ones where there was no calls. And so if you were seeing some progeny where there were a lot of missed calls, no calls, you might want to consider removing those, because maybe they're not that good. So now we want to calculate the co-segregating SNPs and remove these identical ones. And so that was done, and so you can see them sorted here. And And if you look in this column here, it says similarity. So all the ones you're seeing on this screenshot right now are all ones that were at the same location, mapped to the same bin. So I think this is kind of a point I'll bring up right now, is that we had a lot of SNPs, but we only could map so many because in an F1 cross here, you only have so many recombination events with progeny size of 92. So if we want to see more SNPs mapped, we need a larger population size. But we also be nice is if we had more recombination going on, like in some of the self-pollinated crops where you can develop recombinant inbreds, so you can reduce these linkage blocks. So now another tool that's in here is you can look at the similarity of the progeny. And so we have two progeny here, or two examples. So progeny 45 and 47 had a similarity of 1. So I think we removed that. Is that what, Joe? So we pulled this one out of the population for the analysis because they had identical similarity. I don't know, so for all the SNPs we looked at, they were identical. Whether they are identical or not, I'm not really sure. Okay, and then there was a second one that was almost 9. It was 99% similarity there. So, but we kept that one in. So now, this is the process of generating the map. And so what we need to do is calculate the groupings. And so what this information gets generated when you run your file in the join map. And what you're seeing here is the different iterations. And what we're looking at is the log scores and the number of markers that fit into that grouping. And highlighted in red are the 12 linkage groups that we chose. So it's not completely clear cut. You have to kind of make some decisions yourself on what groupings you're going to use to generate your linkage groups. So hopefully you should get 12 when this is all done. Okay, so now we go ahead and create the linkage, create the map for the linkage groups, and so you can see here now we have the different SNP locus, and then you have the position, and you can see along the chromosome starting from 0, and it continues on past 51 centimorgans. And then, which you can then take that data file and create the map output for each linkage group. And this is what you'll see. Oops, that's what you're used to seeing. But right now, we don't know which linkage, which chromosome this is associated with. So now, what you can do is go back and look at the SNPs that you have, go back to the pseudomolecule, and make and then call that the actual chromosome. And so that's what we have here now is we have chromosomes 1 to 6, associated with the linkage groups, and here from 7 to 12. So that's the process that we go through. So we've made the map, and so we align them with the pseudomolecules. And what we need to do is look for mismatches now. Okay, and take a minute and talk about that. It could be that some of these mismatches might be because it's a bad assay, or it could be that there's a paralog, that maybe there's this SNP maps to more than one location. And so here's some examples of mismatches. So the first one here, this looks nice. So if it's a mismatch and it looks right like this, so maybe the pseudomolecule has it in the wrong place. And so maybe this is where the concordance between the map, the genetic map and the physical map can be corrected. This is a situation where we're looking at the graph here, and we know that the heterozygotes should map to the, or should show up in the middle here, and you can see the circle around this is saying that, yeah, they're all heterozygotes, but if you look at them, they're clustering into different groups. And so I'm not going to go into the details of it, but we've sat around and kind of did some thinking through this, and if there's a paralog, there's going to be some intensity differences, and also which may actually influence some of the segregation differences, and so this may be a clue that we have a paralog. So if you see something like that, you want to be wary of what's going on. And then here we have a mismatched SNP, but what you have really is a messy data set. So I think this is one where I would go out and throw this one away and not use that. So here's a table that's summarizing the SNPs that were mapped in the two diploid populations. We have the pseudomolecule, and so there were, by chromosome, we had 133 in the DRH population that were not mapped in the pseudomolecule, and we had 232 that were not mapped in the D84 population. So of those ones that weren't mapped, we had mismatches, as you can see, by chromosome, and you can see that on chromosome 10, we've seemed to have some large numbers of ones that were kind of mismatched. But then there's a scattering around. So we got the 10 there, and 9 on chromosome 12. So so by having the two populations here, I think that kind of helps us have a little more confidence on where whether it's something specific to the chromosome or whether it's just that, as Robin said, we got a lot of things right in the pseudomolecule, but there's maybe some orientation issues and things like that. But I think this is kind of helpful in the process. I'm going to stop here at the moment because I think this is kind of the process of going through the map, and I want to talk a little bit about tetraploid SNPs, but I think this is kind of take a couple minutes stopping point here. If people got questions about the diploid mapping, any comments or anything along the way? Walter? So Walter's asking about the cost of the programs. Join map I think is a little pricey. And so we have so Dave is saying about $4,000 for join map.
Hey Dave, I got a question.
Okay, so Walter's asking about map QTL. Dave, what do you have so it's about $4,000 for the programs.
Hey Dave, this is John. I got a question from online.
Okay, I got a question from online.
What do you mean by not mapped in PM?
So the question is, what about being not mapped in the pseudomolecule? Okay, so the pseudomolecule, there was a lot of SNPs were chosen based upon their location in the pseudomolecule, but I guess some were not, when it was all said and done, didn't have a chromosome assigned to them. And so in our data file, they were unassigned. But we still used them because we felt they were good SNPs for various reasons. So Dave Francis is saying there could be in some gaps in the sequence there. Robin, do you have a comment on that? So I can't completely summarize what Robin was saying there, but there was just gaps in some of the sequencing because they're coming from some of the transcriptome work and between some of the scaffold work. So I think it just goes back to the point that the genome sequence does have some gaps, and this is helping to fill some of those in. Any other questions that you're in, John?
I had one that was actually from Robin's talk that I just missed, and someone asked about the what about the condition that SNPs had to be at least 50 base pairs apart? If you don't want to take that now, it's okay, but that was asked.
We'll come back to your that's an Infinium. Well, actually, it's just an Infinium design question answer. Is that so? You're asking whether this why do the SNPs have to be 50 base pairs? Because the primers used, I think, are require that spacing around there so that we don't have any mismatches in the along the sequence data. If we have too many mismatches, we're not going to have the primers annealing to the DNA sequence.
Thanks.
Any other comments or questions at this point?
I got one more actually.
John, hang on 1 minute.
So the question has come up about the cost of some software involved. And I think what users have to understand is that you're not locked in to join map, as an example. There are other software packages that can be used in map that's still used for this. One of the obstacles is grouping as many markers as you have. Right? So if people aren't going to use join map, they may want to consider creating first scaffolds, grouping those based on what we know about the genome sequence, and then adding markers. If you're working with a service provider, they may just give you the Genome Studio file,
Right.
essentially an Excel format, but you still need to go through with your own criteria in terms of eliminating progeny that don't have enough SNPs on them or markers that have too much missing data. So the step-by-step process that you've articulated is appropriate for any software that a user might choose to go with. So see if we can repeat all of that.
Yes. We really needed a microphone for the audience here. That's a point to make for next year if we're doing this. But so Dave Francis was commenting about really not being locked into using join map software. There's lots of other softwares out there that people can actually use or purchase, and there's but you still need to be able to manipulate your well, you got to go ahead and filter your data before you do any of the mapping work. Is that what you're?
So the point I'm making, though, is if you're working with a service provider,
Okay.
then you could work directly with the Excel file that you get from them.
Yeah.
But you still have to then go through and do some quality control and throw out some more.
Yeah. So if you're working with a service provider, you know, like there are some private companies that are doing the genotyping that you're going to have to go through and still go through and filter these out the data like we're going to here. Is that what? So the Genome Studio software I don't think is outrageously priced. Dave Francis is saying it's about 2,000, right? Maybe even be 1,500 or something in that range. We purchased a few site licenses for Michigan State, so we have a few copies on our campus. Okay, so I'm just going to take a few minutes just to talk about tetraploid SNPs. I thought we would be further along at this point, but it's taken us a lot longer to move through this, the diploid analysis. But I feel that we've gotten it right to this point. And so during the week, I have a talk where I go into more of the details of those two diploid maps. And so I'll presenting more of the data. I was kind of doing more of the process of walking through this right now for you as the mechanics of it. So as potato breeders, we really want to be able to use tetraploid populations, and so does this really? How do the SNPs behave in the tetraploid material? And so we're using the Illumina platform, and we're using the Genome Studio software, and so we're using this iScan system, and they call it microarray based, but there's a bead express system, and I'm going to come back come back to that. So there's other platforms within Illumina that also can be used that don't require you to, well, you can use a less number of SNPs, ones that maybe are only important to you. Okay, so with the tetraploid, we got it gets a little more complicated because we have 5 marker classes, but then there's also this question about if we have any nulls and how does that play into that. But when we're working with the diploids, we really just have the 3 clusters. So Candy-Hansey, in looking at the data coming out of SOLCAP, we have the russet mapping population, plus we also have some tetraploid genotypes in the panel, has been able to look at the data from a tetraploid point of view. And so here is a nice situation where we have the 5 good clusters to work with. I just want to point out here that you can see what happens is that the Genome Studio software kind of puts a circle around the different clusters. And so if they fall outside of that, those are the ones that you want to exclude from your analysis, okay, or consider excluding. And so when you're dealing with the tetraploids, you have all different types of segregating or segregating progeny we can have here. We're going to have the 5 different genotypic classes, and then you have different complex segregation ratios. You have the 1 to 4 to 1 up here, and then you have the 1 to 8 to 18 to 8 to 1. And so I think even the potato breeders sitting in the room here, if we ask you to kind of calculate that out, might get stuck for a few minutes trying to work your way through that. So looking at our data here, that Candy feels that these are some ideal SNP markers in terms of the way that they're sorting into the different genotypic classes. So you got to be really careful on handling the data. So in the diploids, everything was based on a 3 cluster call I told you about. Candy going through SNP by SNP, and so she's also doing it now from a tetraploid point of view. And so what you can see here is these have been screenshots from the Genome Studio, and so what she did was she was color coding within the full panel of material that she was working with, the tetraploid tetraploid russet mapping population. And so here we're expecting the 1 to 4 to 1 segregation ratio when we have a duplex parent cross the homozygote, and you can see them falling into those categories. And then over here in the other screenshot, we had a duplex parent cross to the other duplex parent, and you can see them falling into the 1 to 8 to 18 to 8 to 1 segregation pattern. So this is, to me, very exciting that we can have this type of segregation data. So when you work through this 5 cluster calling, so Candy's still working on it, on the QC, but this is what she's saying based upon the theta values. Remember I talked about those theta values in the Genome Studio, and so 2,645 fall into 5 clusters, and 858 fall into the 4 clusters, and so on. So there are a number of SNPs that are going to be useful in being able to call at this 5 genotypic class. But I think you want to go and look at them yourself in your populations there. So okay, so we want to be able to, now that we're kind of wrapping up this diploid, we have the russet mapping population. We have some other tetraploid populations. So we're going to start to look at that and move to the tetraploid data, and we'll have some advice on that in the future. It's getting into harvest season, so it could get delayed a little bit. So just as a kind of a reiteration, this is what we said the SOLCAP objectives were a few years ago. And so we wanted to make up to 10,000 markers and associate these markers with our carbohydrate genes, which are important in the processing industry. And so we've been able to make a SNP chip. There's 8,300 SNPs on there. So I think we're in that range. We wanted to collect standardized phenotypic data over multiple locations. Walter was reporting on that today, so I think we're moving forward. And let's not forget our tomato friends here. This project we're focusing today on the potato, but really I think what's helped driven this project forward is actually working with our colleagues in tomato breeding and genetics. I think having that expertise, I think, has helped us on the potato side. And I think the tomato people appreciate that they don't have tetraploids. So, and then we talked about having other populations that would be SNP genotype. And so I showed that slide earlier that we have those different populations that have been submitted by the community. And so one of the questions I could put out to the people is, are there more populations out there that we need to consider that would be valuable to the community? Right now, the ones I have listed there, and even some others, we've got so much data to work with. We're going to keep ourselves quite busy. But I think this is, the bottom line right here is that we need to create this integrated breeder-focused resources, and so these databases and browsers that are developed. What Robin talked about, with her browser, I think is really critical. I kind of feel as a potato breeder, I got to be able to access that browser and use that information now that we have the genome sequence, and use that in our genetic analysis, and with the data, with the phenotypic data, with the SNP data available, these are going to be up there. As a breeder, how can I use that data? How is this going to help my breeding program move forward? So what we feel that we've got these resources. We've got the cDNA libraries, the high throughput SNP analysis, the germplasm panels. We've got the SNP chips. We've been running these workshops. The whole point of the workshops is to help you feel more comfortable of where things are going, and we have the databases. And so what's going on in 2011? We've been SNP genotyping. All the obligations on the potato side for SOLCAP have been done, and now we're just dealing with ones that people have submitted for their own purchased chips. We're working on the databases, and we're working on the publications. We're going to move to the QTL analyses, and we want to have a hands-on workshop to help people work through things. And also, we have the eextension.org site. Maybe when Dave Francis is talking, he can just take a minute and kind of update people on that. And so this is a place where also you should be able to find some of this information that might help you move forward. It should be a resource for the breeders, not just the potato breeding and tomato breeding community, but the breeders in general, where you can find information that would be helpful to you. So I think you can kind of capture it in this one line here, is that what's the outcome of SOLCAP is a genome-wide set of markers and bioinformatic tools accessible by the breeders. So we're moving towards that accessibility. I think the tools are being developed, and I'm quite excited. And I'm just going to, this is my last slide here, and I'm kind of feeling like, okay, where does this all go with the, with this analysis? I remember we were saying, well, after we develop some SNPs, we'll convert them back to PCR-based agarose gel markers that you could use. Well, I think that's kind of silly. You know, I really, the high throughput analysis is so much more cost effective. And so there's different platforms. And so Illumina Beat Express, you know, they have a you know, platform, and you can generate, you can have a 48 SNP system, a 96, all the way up to 384 that you can analyze. You know, you can multiplex, analyze at one time. That's what we want to do. We don't need to sequentially run all our markers that we need. We can run them in a few days, get our data. So you can see how they got it set up. If you want to buy 480 samples or almost 1,500 samples, what the cost is, right? So, and what this is, I think this is the column to focus on, is what's the price per sample to do the SNP genotyping. So as a breeder, I really don't know how many markers, when this soul cap is all done, that we're going to think are important. And maybe as a breeder, you may think that yourself that you might say, oh, these are the ones that I think are important, or we can do it collectively as a community. But if you say that we have 192 SNPs are important to us as a community, you know, if we wanted to genotype progeny in our program, it's a little over $16 a sample for the reagents. There'll be a small, I think, genotyping cost, but the reagents are going to cost you there. So if you had 1,000 progeny that you wanted to SNP genotype, and so for a little over $16,000, you can do that. Well, and so the question is, will this help move your program forward? And I'm kind of feeling bullish about that there's going to be ways that we can do this. I think this is the exciting part, is where we're going to go with these markers. Can we create a breeder chip, or do we individually create our own breeder chips? But we have that option to do whatever we want. And our SNP chip was based on those 8,300, and you can actually, Robin mentioned that there's 69,000 out there, you know, that fit the Infinium design. So you can go back to those and if you go into the browser, you can find them in there, and maybe those SNPs might be more valuable to you than the ones that we actually use. So this is how you have to start thinking. You know, you can go back, the information's there, and you know, how can you best use this? So I think we're entering kind of an exciting time, and I'm really curious. Like, we could sit around, drink a beer, and say, what's going to happen in the future with the SNPs? So I think, and I'll just finish with the acknowledgments. I really mentioned some of those people. This is really a teamwork to move forward with all this. You know, I mentioned the people, and Robin in my program, that have worked on this, but it's really the team itself. I think if we didn't have our friends on the tomato side, I don't think we would also be there too. It's a great effort that's been going on. So I'll stop at this point and take a second set of questions, if there's any at this point. Paul.
When you say going through the individual SNPs to look for, are they segregating as 5, are they good SNPs, but you're seeing 5 categories in that. Is that really visually looking at every SNP?
Right. Okay, so Paul's asking if whether on those calls on each SNP, yes, it's actually an individual person's actually going SNP by SNP looking at that. So in other words, you get the Genome Studio auto call, but then what you want to do is just take a look at it and kind of agree, or you can change the parameters a little so that it's on what it's including in that call. Dave? So Dave Francis is pointing out that there is a cluster file, so you can go to that, and so you don't have to reinvent that and see what. So that. So Robin, the 3 cluster call is up on the site there, and the 5 cluster call will be up when it's finalized. And there's also, I think, one that's going to be going up for tomato also, right? So.
Online question, Dave?
We have an online question?
Yeah, from Gene Jacobson, New Zealand. asking again about those the PMs. Asking if the data is available, and especially the data not mapped in PM but assigned to linkage groups.
So so Jeannie Jacobs from New Zealand is asking whether those ones that haven't been assigned to chromosomes are going to be, so will the data be available on those, is what you're saying?
Right.
So we're in the process of writing the paper on that, and so I think that'll be coming out in the publications that we'll be submitting shortly.
Great, and she just typed in again to help improve the pseudomolecules, that comment.
Yeah, so I think the idea is to add that to the pseudomolecules. Raman, do you have a comment on that? Okay.
And
so that's where we are at this point.
Great, and one other quick one, you might have answered this one, but question is, are the mapping populations and maps going to be published so they can use the information?
So are the mapping populations maps going to be made available? So I expect that the maps will end up on SGN when, and SOLCAP when as we finalize them there and submit our publications.
Great, that's it for now.
Oh, the populations, yeah, they can be put, they will be put on there. Well, the rest of the mapping population is on there right now, not the SNP data yet, but it will be. So Dave Francis is asking, can somebody get tubers for the populations? I think that you have to go to the individual person on that. And potatoes, that could be very difficult because of the challenge of maintaining a population and keeping the virus out of it. So that's going to come down to individual. I think we're talking about the mapping population and putting one of them into tissue culture. Richard, is your population in tissue culture? So, okay. So DM by RH is going into tissue culture, and we are going to move the DM by 84 SD22 into tissue culture also.